So this is the last and the third stage, and it is known as the MAC, or membrane attack complex. MAC, or membrane attack complex, the MAC, which is a unique system that builds up a lipophilic complex in cell membranes from several plasma proteins. And when fully assembled in the correct proportions, take note, C7, C7, C6, C5b, C5b here, and C8, we need C8, of course, C6, C5b, C7, C8, C8 form the MAC. And then the C5b C6 complex is hydrophilic, but with the addition of C7, C7, it adds up. There is something because C7, it has additional detergent and phospholipid binding properties as well. The presence of hydrophobic and hydrophilic groups within the same complex the complex of MAC may account for its tendency to polymerize, and this polymerization will lead to the formation of small protein micelles, which are packets of chain molecules in parallel arrangement. And then it can attach to any lipid bilayer within its effective diffusion radius, which produces the phenomenon of reactive lysis on innocent so-called bystander cells, or some would say bystander cell lysis. The C5b, C6, C7, C8 complex polymerizes, and then it polymerizes to C9 to form a tubule, and this once C9 is included here, this polymerized complex with C9 forms a tubule. There is a tubule here, a tubule, and this tubule is somehow like a pore, or likened to a pore, which spans the membrane of the cell being attacked, allowing ions to flow freely between the cellular interior and exterior, so in and out of ions. Later, I'll explain why it's deadly to the cell. It can lead to lysis. By complexing with C9, the osmotic cytolytic reaction is accelerated. This tubule is a hollow cylinder. It's a hollow cylinder. With one end inserted into the lipid bilayer, the other end, and the other projecting from the membrane. A structure of this form can be assumed to it disturbs the lipid bilayer sufficiently to permit the free exchange of ions and water molecules across the membrane. Ions flow out, but large molecules stay in, causing water to flood into the cell. So water gets inside the cell. And what's the consequence of this is that in a living cell, it can result to the influx of sodium ions and water. So sodium ions and water molecules, can gain access, and it will lead to the disruption of the osmotic balance, which produces cellular lysis. So the endpoint for MAC, cellular lysis. So now we are on the alternative pathway of how this one is being initiated or triggered. So pathogens that can be destroyed in the absence of antibody by the means of alternative pathway, which acts as a part of the innate or natural pathway. So is it a question? No. Pathogens can be destroyed in the absence of antibodies through the alternative pathway. This one is antibody dependent or independent. It's antibody independent. In the absence of antibodies, pathogens can be destroyed with your alternative pathway. And this pathway is very essential as an early defense against pathogens or microbes. Phylogenetically, this represents the oldest of the three activating pathways. When I was in third year, I thought the oldest was the classical, but it's not. This alternative pathway, this pathway is way back more, is older. If we will compare it with the rest of the pathways, it's phylogenetically older. It's the oldest of the three activating pathways, and it was originally named for the protein properdin, because before it's not known as alternative pathway, this is known as the properdin pathway. However, properdin, remember, the scientists were able to know now that properdin does not initiate this pathway, but rather stabilizes the C3 convertase formed from the activation of other factors. And then soluble C3 in plasma undergoes slow spontaneous hydrolysis of its internal thioester bond, which leads to the formation of a fluid phase C3 convertase here. So it forms again, it leads to the formation of a fluid phase C3 convertase and the generation of C3b. So now we can have again you might for us to remember that soluble C3, there's a spontaneous cleavage of C3. It undergoes slow spontaneous hydrolysis, cleavage through hydrolysis, which leads to the formation of a fluid phase C3 convertase. It's not there, and the generation of C3b. If the C3b is deposited on the surfaces of microbes, so in the microbial surface, the C3b is deposited, it binds factor B. It binds a factor B in here, this one. It binds to factor B and forms the alternative pathway C3 convertase. So this one, it C3b plus binding to factor B leads to the development of C3 convertase. This C3 convertase is stabilized by property. This convertase cleaves C3 to produce more C3b. So the C3 convertase, it will produce more C3b. It cleaves C3. So C3 convertase cleaves the C3 to produce more C3b. And the C3b binds to the microbial surface and participates in the formation of a C5 convertase. Again, it participates in the formation of a C5 convertase. Those again the sorry, those C3bs formed after the cleavage of C3 by the C3 convertase. Again, C3 convertase cleaves C3 and the cleavage products are the C3bs. These C3bs will be deposited and they will they will bind to the microbial surface and then they will participate in the formation of a C5 convertase. And this C5 convertase cleaves C5 to generate it cleaves C5 to generate C5b, the initiating event in the late steps of complement activation. Remember, in the presence of C5b initiates the formation of the MAC starting from C6, 7, 8 through 9. And then again, what are the things we need to remember? Alternative pathway, destruction of pathogens without antibody, so antibody independent, and this one is our defense against an important early defense against pathogens, and it is the oldest of all the pathways and was originally named as Properdin pathway. And now we have the lectin pathway. Please note that the alternative and the lectin pathways provide antibody-independent innate immunity. The lectin pathway is activated by microbial carbohydrates, and the lectin pathway differs from the classical pathway only in the initial recognition and activation steps. Indeed, it can be argued that the lectin pathway should not be considered a separate pathway. Some scientists say that, but rather a route for classical pathway activation that bypasses the need for antibody. The C1 complex is replaced by a structurally similar multimolecular complex comprising a C1q-like recognition unit, the MBL. This MBL the MBL is structurally similar to the C1 complex or C1q complex because it has a C1q-like recognition unit. So MBL or the ficalin or ficalin, they are somehow also having the same structural similarity to C1q, but according to the book, the MBL is really having a structural similarity to the C1 complex. So that's it. So again, the lectin pathway is triggered in the absence of an antibody. No antibodies again. And then initiated by the binding of microbial polysaccharides to circulating lectins. So, because we have the mannose binding lectin, this lectin will bind to the mannose, mannan, or the lectin residues or MBLs and to ficolins. And MBL binds to, again, see, MBL binds to mannose residues on polysaccharides. Again, antibody independent for lectin. So this one, this is the mannose or mannan binding lectin and ficolins. These soluble lectins are collagen-like proteins that structurally resemble C1q. This is the thing I have told earlier, but I'm having a hard way to say it. So again, the ficolins and the MBLs, somehow they have they structurally resemble C1q in the classical pathway. MBL, L-ficolin, and H-ficolin are plasma proteins, and M-ficolin is mainly secreted by activated macrophages in tissues. So I just like to point that this one is from the classical, and this one, these two are from the lectin pathways. And they share somehow, they have a structural homology. Next will be a complement production regulatory system. So control of the complement system is needed. Why is it needed? Why we need to control it? Why we need to keep things in check? Is to prevent the consumption of components through unregulated amplification, and of course to protect the host. Complement activation poses a potential threat to host cells because it could lead to cell opsonization or even lysis. And to defend against this threat, a family of regulators has evolved alongside. In the complement system, we need to really defend it through regulatory system is to prevent or to inhibit uncontrolled activation, and again to protect cells from attack. So we have the following: C1 inhibitor controls the classical and lectin. So some books would abbreviate this one as C1INH. So C1 inhibitor controls for the classical and the lectin pathways. C3, C5 convertase activities are controlled by the DAF, the decay accelerating factor, and enzymatic degradation. And control of the convertases is mediated into complementary pathways. We have decay, acceleration, and cofactor activity. So we'll go over with them one by one. So the C1 inhibitor controls the classical and lectin. So in the activation pathways, the regulators target the enzymes that drive amplification. So activated C1 is controlled by a plasma serine protease inhibitor serpin termed C1 inhibitor or C1INH, or some books C1 capital INH. They're just the same. C1INH, which removes C1R and C1S from the complex, so thereby switching off classical pathway activation. C1 inhibitor also regulates the lectin, because this one is for the classical. It switches off the classical pathway and regulates the lectin. And how does it regulate the lectin pathway? In a similar manner, by removing the MASP2 enzyme from the MBL or ficolin complex to switch off activation. But those things are too detailed to be remembered. So for us to easily remember, C1 inhibitor switches off the classical pathway of activation, and also it regulates the lectin pathway. So here we have the C3 and C5 convertase enzymes. They are heavily policed with plasma and cell membrane inhibitors to control activation. So we have in the plasma and on the membrane. So in the plasma, factor H and related protein factor H like 1, FHL-1 can destroy the convertase enzymes of the alternative pathway. We also have C4 binding protein. C4BP performs the same task in the classical, however, in the classical pathway, same way, but in the classical pathway. And on the membranes, cell membranes, two proteins, membrane cofactor protein MCP and decay accelerating factor DAF, collaborate to destroy the convertases of both classical and alternative. And now we have the control of the convertases is mediated in two complementary ways. So one, we have decay acceleration. So we have if factor H like 1 and C4BP in the fluid phase and DAF. And this, they bind the convertase complex and accelerate decay, displacing C2A from the classical pathway convertase, BB from the alternative pathway. So somehow classical and alternative. We also have cofactor activity, factor I. In the presence of an appropriate cofactor, it can cleave irreversibly inactivate C4B and C3B, while MCP is a cofactor for factor I cleavage of both C4B and C3B. So C4B specifically catalyzes cleavage of C4B, and factor H and factor H like 1 catalyzes cleavage of C3B. A bit difficult to remember, but the main thing you need to know that these cofactor activity, they need cofactors to elicit their effects to control the convertases. Because C3 convertase, something like that. See? C4B, C3B, cleavage of C4, and that cleavage can lead to control. And we also have properdine, which is a unique positive regulator of the alternative pathway because it can stabilize. Please remember this one, very important. Stabilize the function of properdine is to stabilize C3 convertase. And thereby, what it gives that stabilization somehow gives the C3 convertase a long lifespan, right? Increases its lifespan. We also have the CR1 or the complement receptor 1. It is a membrane regulator of C3 convertase activity and a powerful regulator with both decay accelerating and cofactor activities in both pathways. So now we are on the part on disease states and complement. So guys, although complement acts as a powerful weapon to combat infection by amplifying phagocytosis, in some cases it can actually contribute to tissue damage or death. Complement can be, take note, it can be harmful if it is activated systemically on a large scale, as in a gram-negative septicemia. Activation of the complement on a large scale is not good. So there is this gram-negative, especially in cases of gram-negative septicemia. It is activated by tissue necrosis, such as in acute myocardial infection or lysis of RBCs or erythrocyte lysis. So these are the scenarios in which complement activation is not okay. So in the case of septicemia, due to a gram-negative organism, large quantities of C3A and C5A are generated, leading to there is neutrophil aggregation, and clotting and damage to the tiny pulmonary capillaries, and interstitial pulmonary edema may result in. We also have tissue injury following obstruction of the blood supply. Here, obstruction of the blood supply, such as those that happen in acute myocardial infarction or heart attack, can cause complement activation and the deposition of membrane attack complexes on cell surfaces. So receptors for C3A and C5B have been found on coronary plaques, including that complement components may increase the damage to heart tissue. So that's it. Aside in gram-negative septicemia, we can observe C3A, C5B in tissue injuries brought about by the obstruction of blood supply. We can also see the presence of C3A and C5A. And next, lysis. RBC lysis. So lysis may be another end result of complement activation. Hemolytic diseases such as called autoimmune hemolytic anemia are characterized by the presence of autoantibody. Those antibodies that bind at low temperatures. When these cells warm up, complement fixation results in lysis. Okay. So now let's go to the major pathway component deficiencies. So although excess activation of the complement system can result in disease states, lack of individual components also has a deleterious effect. So hindi lang yung marami ang problema, pero pag nagkulang problema pa rin. So, hereditary deficiency of any complement protein with the exception of C9 because take note guys, C9 doesn't have any known disease association. Walang problema kung maraming C9 or walang C9 kasi it's not associated with any disease process. And then again, deficiencies, hereditary deficiencies ng kahit ano of any complement protein usually manifests itself in increased susceptibility to infection talaga yan. So, madali kang magkasakit, increased susceptibility to infections and delayed clearance of those immune complexes, yung mga C1Q natin na, di ba, saan na napunta. Most of these conditions are inherited on an autosomal recessive gene and the deficiency that happens most often is that of C2. So, yung pinaka-common, most commonly occurring deficiency is C2 deficiency. So, the presence of that deficiency will lead to the development of atherosclerosis, recurrent, palagi na lang nagkakaroon ng streptococcal and staphylococcal infection. So, the C2 deficient individuals may be more prone to recurrent streptococcal and staphylococcal infections. And the factor B locus is nearby and often C2 deficient persons are reported to have decreases in factor B also. Other types of complement deficiencies are less common. Pero basta yung pinaka-common C2. Pero walang problema yan, di naman masyad. The most serious of all the deficiencies will be that of the C3 deficiency. The most serious deficiency is that of C3 because it is the key mediator in all pathways. Again, bakit serious yan? It is the key mediator in all pathways. Individuals with a C3 deficiency are prone to develop severe, take note, severe recurrent, paulit-ulit, life-threatening infections with encapsulated bacteria. Such as what? Streptococcus pneumoniae, and may also be subject to immune complex diseases. Bakit? Yung immune complexes problema ba? Yes, such complexes can be lodged in the kidneys and result in AGN or glomerulonephritis, acute glomerulonephritis. So, yun ang problema. Again, so, bakit yung C3 deficiency is the most serious? Kasi C3 is the key mediator in all pathways. And then, the C3 deficient individual is highly prone to develop severe recurrent life-threatening infections with encapsulated bacteria. So, ano yung mga bacteria na iyon? Streptococcus pneumoniae, number 1 example. And pwede din magkakaproblema sa formation, maraming immune complex formation. So, may immune complex diseases. Kasi antigen-antibody immune complexes can get lodged into the basement glomeruli of the kidneys, diba? And it appears that the deficiency of any of the terminal components of the complement cascade, like C5, 6, 7, 8, causes the one which is not here is deficiency ng kahit like again, ano ba yung sinabi ko? any deficiency of any of the terminal components of the complement cascade ng C5, C6, 7, and 9 causes increased susceptibility to systemic Neisseria infections, including meningococcal meningitis and DIC or disseminated gonorrheal diseases. So this one, if we would want to tabulate what are the first column, we have the deficient component. Second column showing the diseases associated with that deficient component. So C1 and C2, lupus-like syndromes and recurrent infections. And it's easy. We will just try to group them together. And how about PNH? The deficiencies of DAF and MERL. MERL is also known as CD59. And we have for the properdine, Neisseria, and for the MAC, we also have Neisseria infections. So for the complement deficiencies, regulatory factor components, let's have them one by one. DAF deficiency. DAF deficient erythrocytes are prone to lysis by means of the bystander effect. Bakit? This is a deficiency in the glycophospholipid anchor or the glycophospholipid inositol anchor of the DAF molecule, preventing its insertion into the cell membrane. So yung DAF, the DAF molecule cannot insert into the cell membrane because the individual doesn't have the anchor for that DAF molecule. And anong problema nito? Complement-mediated intravascular and extravascular hemolysis leading to chronic hemolytic anemia. In fact, this is a urine sample from a person with paroxysmal nocturnal hemoglobinuria. See? And the DAF deficiency is highly associated with the lack of CD59 or MERL. Diba? Walang ako, walang Miss MERL, hindi. Walang CD59 or MERL, and both are implicated in PNH. Kasi ano yung MERL? CD59, also as known as CD59, has the same glycophospholipid anchor found in DAF. So the gene deficiency affects both molecules. So DAF deficiency, also walang CD59. Anong resulta? PNH, paroxysmal nocturnal hemoglobinuria. And then, meron din tayong the lack or the deficiency of C1 inhibitor, C1 INH. So, it will lead to hereditary angioedema. So, recurrent attacks of angioedema that affects the extremities, the skin, the GIT, and other mucosal surfaces. Other mucosal surfaces are really characteristic of hereditary angioedema. And this disease, ang disease na ito is caused by a deficiency or the lack of C1 inhibitor, which happens or occurs with a population frequency of 2 in 10,000. So, lack of C1H results in excess cleavage of C4 and C2. So, maraming C4. Excess cleavage, nakikleave. C4 is broken down, C2 is broken down, keeping the classical pathway going and creating kinin-related proteins. So, bakit ba? Kung maraming kinin-related proteins, kasi these proteins increase vascular permeability. Thus, the response to a localized antigen is continued swelling, or edema. Diba? The edema can be subcutaneous or within the bowel or upper respiratory tract. Normally, this spontaneously subsides, nawawala din naman in 48 to 72 hours. But if this occurs in the area of the oropharynx, life-threatening upper respiratory obstruction may develop. So, still, hindi maganda. Now, we are on the laboratory detection of complement abnormalities. So, determining the levels of complement components can be a useful adjunct in diagnosing disease really. And hereditary deficiencies can be identified, and much can be learned about inflammatory or autoimmune states by the following consumption of complement proteins. So techniques to determine complement abnormalities generally fall into two categories. We have two categories. So we have measurement of components as antigens in serum, measure natin sa serum, or the measurement of their functional activities. However, many assays are not available in routine clinical laboratories. Wala tayo niyan. And they are just restricted to research laboratories. That's the sad thing. So we have immunologic assays for individual components. Most frequently used to assay individual components. Ano bang nangyari sa slide na ito? Basta yung pinaka-commonly utilized or used to assay individual components ng like complement fragment na like we would say C3, C1, C2. We will use RID or radial immunodiffusion, and we also have the other one which is known as nephelometry. Don't worry, I will be explaining the principles underlying such assays pag under labeled immunoassays na yung topic natin. Okay? So, overall complement activity measurements. So, overall, global. So, hemolytic titration assays, we have may dalawa tayo, CH50 and AH50 test. Ano ba yung CH? Ano ba yung AH? C stands for classical, A for alternative, H for hemolytic. 50 kasi 50% how much of it in the serum can lies 50% of the synthesized or of the synthesized sheep RBCs. So again, ano yung C? C for classical, A for alternative, H for hemolytic, and kasi ano to 50%, and they are utilized to measure global complement activity. So H50 test is for total serum alternative pathway kasi A nga alternative protein measurement, while CH for the classical as measured by the osmotic lysis of sheep RBCs. Oh. So ito, we have the assays for the classical pathway, CH50 test. Classical ito. Hemolytic titration CH50 assay, ito ang pinaka-common. This is the most commonly utilized assay that measure lysis on sheep red blood cells. Paano ba? Ano ba? Anong nangyayari dito? It measures the amount of patient serum required to lyse 50% of a standardized concentration of antibody synthesized sheep RBCs. So, yung kawalan or absence of any, kahit in isang complement component will result in an abnormal CH50, essentially reducing this number to 0. So, meaning pag 0, walang complement component, kulang yon. The higher, the better. The lower, the more like drastic. And then, titer is expressed in CH50 units, which is the reciprocal of the dilution that is able to lyse 50% of the synthesized cells. So, ganun siya. We also have the alternative pathway activation assay, and can be measured by several different means. We have may dalawa. An AH50 assay test can be performed the same manner as the CH50. So, pwedeng same lang naman yung ang style ng H50 sa CH50. Except, may exception siya. Except magnesium chloride and your ethylene, yung EGTA, not EDTA ha, tama talaga to. It's really glycerol, tetraacetic acid. It's really EGTA. Are added to the buffer. It's the except magnesium chloride and EGTA are added to the buffer and calcium is left out. Ano bang meron ng buffer na ito? Because this buffer, anong function? It chelates calcium, which blocks the classical pathway of activation. And my modification naman, rabbit red cells are used as the indicator here because these provide ano ang reason? It provides an ideal surface for alternative pathway activation. I would just like to note kung dito yung ginagamit natin kasi sheep sa CH, sheep RBCs. Here, they are using the rabbit RBCs. So, another one, an additional means of testing for the alternative pathway function is via ELISA or enzyme-linked immunosorbent assay. One such test can detect C3b, BPB, or C3BP complexes in very small quantities. So, yan. In very small, take note. Ganito yung ELISA ay naka 96 microtiter well plate commonly. Microtiter wells are typically coated with the bacterial LPS to trigger the or polysaccharide to trigger the activation of the alternative pathway. Kasi alternative ito. So we have alternative and for H50 and ELISA. Pwede bang mag-ELISA sa CH50? They haven't discovered it yet. So this is the end. So just a reminder for everyone, don't limit yourself, and many people limit themselves to what they think they can do. You can go as far as your mind lets you. What you believe, remember, you can achieve. Thank you. Good day, good night, good dawn, good afternoon, or good noon to all, and sana this can really help in your studies. God bless.